Okay, so picture this. It's the mid-19th century. We are smack in the middle of, probably the most exciting, chaotic scientific revolution in history.
Everyone is arguing about biology, where life comes from.
Exactly. And amidst all this noise, sitting on a shelf in a monastery library in what's now the Czech Republic, there's this thin little booklet. And inside that booklet is the answer.
The answer to one of the biggest mysteries of existence. The literal operating system of life.
The code. And here's the kicker. That booklet sits there gathering dust, completely ignored, for 34 years. It's like having the blueprints for the internet in 1860 and, I don't know, using them as a coaster.
It is arguably the greatest missed connection in the history of science. A real tragedy of timing.
So today we're doing a deep dive into that tragedy, which luckily eventually turned into a triumph. We are talking about a monk, a very specific garden full of pea plants, and a discovery that was just so radical, so far ahead of its time, that the scientific community, they just sort of blinked and moved on.
We are, of course, talking about Gregor Mendel.
Now, I know what you're thinking. Oh, the pea plant guy from high school biology. But I really want to push past that cartoon version of the monk in the garden. This is not just a story about gardening.
No.
This is a story about a man who applied the rules of physics to nature and saw patterns that, well, literally everyone else had missed.
And that's the absolute key. So our mission for this deep dive is to figure out how he did it. How did a monk working in a monastery discover the laws of genetics before anyone even knew what a gene was?
And maybe more importantly, why did he succeed when the greatest minds of his day, people like Darwin, were just hitting a wall?
Exactly.
So let's unpack the man himself first. Gregor Johann Mendel. I think the popular image is this, kindly old man who just liked pottering around the garden, but that's not really right, is it?
Not at all. He was born in 1822 in northern Moravia. And to really get his work, you have to understand his struggle. He was not part of the scientific aristocracy like Darwin was.
Right.
He was the son of a peasant farmer.
So he didn't have the luxury of just thinking for a living.
Far from it. His family were gardeners, beekeepers, so he understood nature on a practical level, but they had very few resources. He was incredibly bright, but the path to education was just too expensive. So his decision to join the Augustinian monks in Brunn, that's modern-day Brno, was largely an economic one.
He joined the monastery to get a scholarship, basically.
In essence, yes.
Yeah.
But we shouldn't think of the monastery as some quiet retreat from the world. The Abbey of St. Thomas in Brunn was a powerhouse, an intellectual hub for research, for agriculture.
So it was the place to be for a smart kid with no money?
Absolutely. And the order saw his potential. They actually invested in him. They sent him to the University of Vienna.
And this is where the story takes a turn, right?
This is the plot twist that changes history. He goes to Vienna, and yeah, he studies botany and plant physiology, but he also studies physics and mathematics. He studies under Christian Doppler.
The Doppler effect guy?
The very same.
That seems like such a weird mix for someone studying plants.
It was completely unheard of, but that is exactly why he cracked the code. When he came back to the monastery garden, he didn't look at his plants like a naturalist, describing things with poetic words.
He looked at them like a physicist.
He looked at them like a physicist. He saw the world in data, in numbers, and statistical laws.
He brought a calculator to a flower show.
Precisely. And in 1856, he begins the experiment. And I want to pause on the scale of this, because it's easy to underestimate the sheer labor involved.
The source material mentions he worked with Pisum sativum, the common pea plant. But this wasn't a weekend project.
No. He was working on a plot of land that was only about 2 hectares. But on that little plot, between 1856 and 1863, he cultivated and tested approximately 28,000 plants.
28,000. And he didn't have a team of grad students helping him out.
It was mostly just him. Sometimes, a little help from his fellow monks. But even before he started the main experiment, he spent 2 whole years just testing his varieties.
What for?
To make sure they were pure breeding.
Meaning, if he had a tall plant, he had to be 100% sure it would only produce tall offspring if he just left it alone.
Exactly. He had to calibrate his instruments before he could run the test. He was incredibly meticulous.
So why peas? Was there something special about them?
Oh, it was a brilliant design choice. Mendel needed a model organism that fit 4 very strict criteria. First, they had to grow fast. You can get a generation in a single year.
Right. If he'd studied oak trees, we'd still be waiting for the results.
We would. Second, lots of offspring. He needed a huge sample size for his statistics to mean anything.
You can't draw a probability curve with 3 seeds.
Exactly. Third, and this is so crucial, he needed varieties with distinct opposing traits. No middle ground.
So, like, tall or dwarf, purple
Again.
flower or white flower.
Yes, binary data. And fourth, he needed to control who was mating with whom. Pea flowers are naturally bisexual, and they're closed, so they self-pollinate.
Which is great for a control group, but Mendel wanted to cross them. He wanted to play matchmaker.
And this is where that gentle monk in the garden image just gets replaced by something closer to a laboratory surgeon.
Right, I was reading about this. The technique he used, emasculation. It sounds incredibly delicate.
It is. So imagine a tiny, unopened flower bed. Before it's mature, before its own pollen is ready, Mendel had to pry it open. And with these little surgical tweezers, he had to remove the stamens.
The male parts.
The male parts, exactly, to make sure that flower could not fertilize itself. He was turning a bisexual flower into a female-only flower.
And he had to do all that without damaging the female part, the stigma.
Yes. And after that, he had to protect it. He'd tie a tiny little bag over the flower.
Bagging.
Bagging. To stop any stray pollen from the wind or insects from getting in. He created a sterile environment in the middle of a garden.
So when he finally came along with the pollen from the male parent he chose, he knew with absolute certainty who the parents were.
No undesirable cross-pollination. He did this thousands and thousands of times. The discipline is, it's staggering.
Okay, so he's got this incredibly rigorous setup. But to really understand why his results were so shocking, we have to talk about what people thought was happening with heredity at the time.
Right. The prevailing wisdom was something called blending inheritance. It's very intuitive. You cross a tall parent and a short parent.
You get a medium child.
Exactly.
Yeah.
Like mixing black paint and white paint to get gray.
Yeah.
It just makes sense.
And even the big guns, like Darwin, were proponents of this.
Oh, absolutely. Darwin proposed a theory called pangenesis.
Yeah.
It's fascinatingly wrong. He believed that every cell in your body gave off these tiny little particles he called gemmules. Gemmules. And these would travel through your bloodstream and collect in your reproductive organs.
So if I, like, worked out and got big muscles, my muscle cells would send big muscle gemmules to my kids.
That was the implication, yeah. It sort of leaned toward the idea that you could inherit acquired characteristics. But the bigger problem for Darwin was that if traits always blend, variation should disappear over time. If everything just mixes down to the average, you eventually just have a gray soup. And evolution needs variation to work. So Darwin was really stuck on this.
And then enter Mendel. He starts crossing his purebred tall plants with his purebred dwarf plants. If blending is right, he should get a bunch of medium plants.
He should. But that is not what happened.
Yeah.
In that first generation, what we now call the F1 generation,
Yeah.
every single plant was tall.
Not medium. Tall.
Not even a little bit medium.
Big.
As tall as the tall parent.
So the dwarf trait just vanished.
It was completely gone, wiped out. So at first, it looks like the dwarf trait is just lost forever.
Mm-hmm.
But Mendel, being the physicist he was, he didn't stop there. He took those F1 tall plants and let them self-pollinate.
He let them breed with each other.
Right. And in the next generation, the F2 generation, the dwarf trait came back.
Get returned from the dead.
And it didn't just come back randomly. This is the moment that changed the biology forever. It came back in a specific, predictable ratio.
The famous 3-to-1 ratio.
For every 3 tall plants, there was 1 dwarf plant. And because he had counted thousands of these plants, he knew this wasn't an accident. This was a law.
A law of nature.
Exactly. So this led Mendel to completely reject blending. He proposed what we call particulate inheritance. He realized heredity is passed down in discrete units, like marbles, not paint. He called these units factors.
Which today we call genes.
We do. But his insight went even deeper. He figured out that these factors come in different versions. We call them alleles. So at a specific spot on a chromosome, a locus, you have an allele for tallness or an allele for dwarfism.
Okay, so let's break down that 3-to-1 ratio because this is really the mathematical core of the whole thing.
Let's do it. So the pure tall parent plant has 2 tall alleles. Let's call it TT. The pure dwarf parent has 2 dwarf alleles, TC.
Makes sense.
When you cross them, the offspring gets 1 allele from each parent. So that first generation, the F1, is all TT.
Got it. One of each. But they all look tall.
Right, because the tall allele is dominant. It masks the effect of the recessive dwarf allele. And this is where the terms get a little technical, but they're so important. That F1 plant is heterozygous. It has 2 different alleles.
It's a hybrid, a mixed bag genetically.
Correct. Now, when that heterozygous plant makes its own pollen, or eggs, half of them get the big T allele and half get the little t allele. It's a coin flip.
So when you cross two of these hybrids,
You're flipping two coins at once, and there are 4 possible combinations. You can get big T from both parents dash TT. You get a big T from one and a little t from the other TT.
or a little t from the first and a big T from the second.
Which is the same thing genetically, TT, or
Yeah.
you could get a little T from both.
You have TT.
Exactly. So now look what you have. The TT plant is obviously tall. That's homozygous dominant.
And the two T plants are also tall because the T is dominant.
Correct. They're heterozygous, but they look tall.
Mm-hmm.
So that's 3 combinations that result in a tall plant.
And the only way to get a short plant is the 4th combination, TT, the homozygous recessive.
That's the only time the recessive trait gets to show itself. So the ratio isn't magic. It's just probability. 3 ways to look tall, 1 way to look short.
3 to 1.
And that was the revolution. Mendel proved that what an organism looks like, its phenotype, can be different from its genetic makeup, its genotype.
This just completely shatters the idea of blending. The dwarf trait wasn't diluted or destroyed. It was just hiding.
It was latent,
Yeah.
waiting for the right mathematical combination to pop back up.
So he has it. He has the answer. He
Yeah.
has the math. He writes it all up in his paper, Experiments on Plant Hybridization. It's February 8th, 1865. He stands up in front of the Brunn Society for Natural Science to present his findings.
This should be the moment the fireworks go off.
Sure.
Champagne corks popping.
But they don't.
No. There was, polite applause.
No.
But the minutes of the meeting show there were no questions, no discussion at all.
Why?
The audience was made up of botanists and naturalists. They were used to descriptive biology, classifying leaf shapes, petal colors, and suddenly this monk is up there talking about algebra and statistical ratios.
He was speaking a completely different language.
Totally. They thought he was just giving a talk about how to breed better peas. They completely missed the universal law he was describing. They didn't get that he was explaining how life itself works.
That is just devastating.
It gets worse. The paper was published in 1866. It was sent to libraries all over Europe. We know it was cited maybe 3 times in the next 35 years.
It just sat there.
It just sat there. And this brings us back to the great tragedy with Darwin.
Right. Darwin was desperate for a working theory of heredity.
He needed it to make natural selection truly work. And the answer, the paper that would have solved his biggest problem, was out there. It was in libraries he and his circle had access to.
So if Darwin had just read that paper,
Genetics would have started in the 1860s, not the 1900s. The great synthesis of evolution and genetics would have happened decades earlier. It's a massive what-if in science history.
but instead, Mendel died in 1884.
He died of kidney disease, respected as the abbot of his monastery, as a meteorologist, but completely unknown as a geneticist. He famously told a friend, "My time will come."
It did, but it took a while.
It took the rest of the world catching up to him. I mean, while Mendel's paper was gathering dust, technology was getting better, microscopes were improving, scientists started staining cells and seeing these weird thread-like things inside the nucleus during cell division.
Chromosomes.
Chromosomes, exactly. So by the late 1890s, biologists knew chromosomes existed, and they suspected they carried hereditary information, but they had no idea what the rules were. They had the hardware, but no software.
And then the year 1900 arrives.
The year of the miracle. In the spring of 1900, within a span of about 2 months, 3 different scientists, all working independently, all rediscovered Mendel's work.
3 of them at basically the same time.
It's unbelievable. It was a photo finish. Hugo de Vries in Holland, Karl Correns in Germany, and Erich von Schermach in Austria. They were all doing their own hybridization experiment.
We're trying to solve the same problem.
And when they analyzed their data and started digging through old literature, they all stumbled upon the same obscure paper from 1866. You can just imagine the feeling thinking you've made this huge breakthrough, only to realize a monk beat you to it by 35 years.
So they didn't really discover it so much as they confirmed it and understood its importance.
Yes, and Karl Correns in Germany really framed it best. He was the one who explicitly connected Mendel's abstract factors to the physical chromosomes they were seeing under the microscope. He saw that the behavior of the factors, segregation, assortment, it perfectly mirrored the dance of the chromosomes during cell division.
That was the missing link. The physical biology finally caught up to the mathematical theory.
And the floodgates just opened. An English biologist named William Bateson read Mendel's paper on a train and became an instant convert. He started spreading the gospel of Mendelism everywhere.
And almost immediately, this wasn't just about peas anymore, was it?
No, that's the power of fundamental law. Very quickly, a scientist named Archibald Garrod applied Mendel's laws to humans. He was studying a rare disease called alkaptonuria. He looked at family trees and realized the disease was inherited in the exact same recessive pattern as Mendel's wrinkled peas.
Wow, that's a massive leap. From a garden in Brno to understanding human genetic disease in just a handful of years.
It proved the universal nature of the discovery. A pea, a fruit fly, a human. The basic rules of transmission are the same.
So as we wrap up this deep dive, we call Mendel the father of genetics, but that title almost feels too small.
I agree. For me, the real takeaway isn't just the genetics, it's the methodology. Mendel represents the moment biology finally grew up and married physics.
It was the shift from just looking and drawing to counting and calculating.
That's it. Mendel succeeded because he simplified the problem. He stripped away all the noise. He didn't try to explain the whole plant at once. He isolated variables one trait at a time, and he trusted the math.
And when the data contradicted the popular theory, he didn't try to fudge the numbers. He followed the data to a new reality.
He had the courage of his convictions, and he had 28,000 data points to back him up. And I guess he had the patience to wait for the world to understand him.
So here's a final thought for you to carry with you. Mendel found the code of life, the looking at something everyone else ignored, the simple binary differences in a common vegetable. He found the signal in the noise because he was willing to count what other people just glanced at.
It really makes you wonder.
What discontinuous variations are sitting in your world right now? What patterns are hiding in plain sight, just waiting for you to stop assuming and actually start counting?
The answer might be on a shelf you haven't looked at in 34 years.
Thanks for diving deep with us.
Until next time.